A groundbreaking study from the University of Utah is offering a remarkable glimpse into what could become the future of heart failure treatment, marking a pivotal moment in regenerative medicine. Scientists have developed a new gene therapy that not only halted the progression of heart failure in large animals, but actually reversed it, something previously thought to be virtually impossible. By restoring levels of a crucial heart protein called CBIN1, the therapy led to a 30% improvement in heart performance, a result that researchers are calling unprecedented. While further studies are needed to determine its safety and effectiveness in humans, the therapy has the potential to transform how we understand and treat one of the world's most deadly chronic diseases. Heart failure is a complex condition, one that affects more than 6 million Americans and is projected to rise sharply as populations age. Until now, treatment has largely focused on symptom management, reducing strain on the heart, managing fluid levels, and trying to slow deterioration. Few therapies offer anything close to repair, and none have provided a cure. This new gene therapy signals a potential shift from maintenance to restoration, and from damage control to actual healing. What makes this treatment so promising is how it targets the root of cellular dysfunction in heart failure. Scientists zeroed in on a scaffolding protein called cardiac bridging integrator 1, or CBIN1, which helps maintain the microscopic structure of heart muscle cells. Patients with heart failure consistently show low levels of this protein, and those with the lowest levels often experience the most severe symptoms and the highest mortality risk. The gene therapy works by delivering a healthy version of the CBIN1 gene directly into heart cells using a harmless viral vector. The result, in preclinical animal models, was nothing short of extraordinary. Hearts began to recover not just in performance, but also in structure. In pigs with advanced heart failure, a model chosen for its similarity to human physiology, those treated with the gene therapy showed significant improvement. All treated animals survived the full 6-month study period, while untreated subjects typically succumbed to the disease within months. The treated animals' hearts pumped more efficiently, their muscle walls regained healthy thickness, and the dilation common in heart failure began to reverse. Even under continued cardiac stress, the hearts maintained their improved performance. This level of reverse remodeling, a return to normal heart size and function, is extremely rare in heart failure research and is typically seen only in the earliest stages of disease, if at all. The therapy's ability to influence so many facets of heart cell function lies in the central role CBIN1 plays. It doesn't just support one protein or pathway, it organizes many of them, acting as a master switchboard for the heart cell's architecture and electrical signaling. By restoring CBIN1, the therapy seems to correct the cellular chaos of heart failure, bringing order back to the tangled network of proteins that power each beat. This kind of central correction is what makes the therapy so powerful. Instead of treating symptoms like fluid retention or high blood pressure, it reactivates the machinery of a healthy heart cell. Importantly, the optimism of the researchers isn't based solely on lab theory or rodent studies. This was a large animal trial, conducted in a species whose cardiovascular system closely mimics our own. That fact alone makes the findings all the more encouraging. While there are still important safety hurdles ahead, such as ensuring the gene delivery virus doesn't cause immune reactions in humans, the path forward is clearer and more hopeful than it has been for decades. There are, of course, reasons for caution. Gene therapy is still a relatively young field, with several approved treatments, but many challenges in delivery, cost, and scalability. Not every patient may respond the same way, and it remains to be seen how this therapy will perform in the context of the wide range of causes and stages of heart failure in human patients. Furthermore, immune resistance to viral vectors, particularly adeno-associated viruses, AAVs, like the one used here, can limit efficacy in people with pre-existing antibodies. Addressing these challenges will be critical to moving from the lab to the clinic. Still, this development opens a powerful and highly promising avenue in cardiac medicine. The idea that a failing heart could be repaired from within using its own cellular components, reprogrammed through genetic delivery, challenges decades of fatalistic thinking in cardiology. It reframes heart failure not as a one-way descent, but as a potentially reversible condition, one that could be met with hope rather than inevitability. The research team, in collaboration with biotech firm Tick&Lev Therapeutics, is working toward human clinical trials, with FDA applications expected later this year. If the data continues to hold, this therapy could be a game changer for millions of people, reducing the burden of hospitalizations, improving quality of life, and extending lifespan in a population that has long needed new options. For now, the scientific world watches closely, encouraged not just by the data, but by the possibility that for the first time, we may be able to restore a broken heart in the most literal sense.